Hello everyone. In this video tutorial, I am going to show you how to use all the functionalities within Spectacle, which is an interactive resource for exploring single-cell gene expression studies in the eye. To access Spectacle, simply open any web browser and navigate to oculargeneexpression.org/single-cell. This is the landing page for Spectacle. There are a variety of menu items on the left-hand side. The first menu item that is automatically selected is the dashboard. This presents the user interface that will be filled in with our interactive plots once we load a dataset. Under Dashboard, we have an About tab that contains some information about our team at the University of Iowa. There's a tab that contains citations and references to all of the included datasets within Spectacle. And next, there's a how-to guide, which contains this video that you're watching and several annotated screenshots that cover how to use salient features of Spectacle. Finally, there's a brief profile on Cell Curator, which is the R package we developed to interact with single-cell datasets, and is freely available on GitHub. Alright, so if we navigate up to our dashboard, we're presented with a drop-down menu for us to load a dataset. Each of these datasets is further described in the Datasets tab on the left. And for this tutorial, I'm going to load a dataset investigating retinal gene expression between a donor with autoimmune retinopathy versus 4 control donors. The first image that appears is the dimensionality reduction plot. Each dot represents the multidimensional transcriptomic profile of an individual cell, and cells are clustered into groups with similar expression profiles. This plot is made fully interactive with Plotly. We can hover over cells and see their cell type, their cluster label, or the library that they originate from. We can also zoom in on certain regions of the plot to look at cells in higher resolution. We can download the plot as a .png image, and this can be useful for manuscripts or preparing PowerPoints. We can also color cells by their originating library. So in this visualization, we can see that each cell is colored according to the library from which it originated. We notice here in the bottom left-hand corner there are 3 groups of cells that are red and predominantly consist of the cells from the periphery of donor 5, which is our autoimmune retinopathy donor where the disease was most prevalent. A major use of Spectacle is to identify what cell populations express a given gene of interest, and there are 2 really good ways of doing this. The first is to use the heat map tab. If you click on the heat map tab, you can scroll down and type in a gene for heat map. So here in this example, I'm interested in the cone-specific gene arrestin-3, and I want to see if this is indeed expressed in our cones. I can select ARR3 from the drop-down menu or hit enter, and within a few seconds, a heat map appears. Cells are colored in this visualization based on their expression of the input gene, with cells colored in a darker blue representing higher expression. I can hover over these cells and identify that indeed cells expressing arrestin-3 are cone photoreceptor cells. We can also visualize gene expression with violin plots. So if I type in the violin plot, very similarly to the heat map arrestin-3, I can view cluster-specific expression for all of our different clusters in this kind of violin format. The neat thing about this plot is I can query multiple genes simultaneously. So for example, I can look at what cell type expresses the gene 1cut2 and see that this is horizontal cells. The nice thing about this is that I can enter as many genes as I want in this visualization simultaneously, and if I'd like to, I can download this plot as a ping. Spectacle also has a recluster function, which allows for potential subgroups of cells to be identified. In this example, I'm going to recluster our bipolar cell populations. On the dimensionality reduction plot on the left, I can see that our bipolar cells are represented by clusters 17, 18, and 19. So I navigate over to the Recluster tab on the right and select clusters 17, 18, and 19 for reclustering. I simply hit the Start button on reclustering, but we're given a warning that reclustering is slow. Reclustering involves renormalizing the expression matrix for all of the included cells. It can take 15 seconds for a very small number of cells and over a minute for a very large number of cells. A progress bar will communicate how far along in the reclustering process the user is, and this progress bar will disappear once the reclustering is completed. So we will patiently wait for the reclustering to finish, and the plot will be visualized in this tab. All right, our reclustering is completed. Well, we can see our reclustered object here, colored by our original cluster labels. So the light blue represents cluster 18, the dark blue represents cluster 17, and the purple represents cluster 19. We can also color cells in a variety of different ways. For example, we can look at potential new subpopulations, so each different color represents a potential subpopulation here. We can analyze cells by what library they originate from, similar to the tab on the left. And finally, we can look at expression of an individual gene that we're interested in. So for example, the gene RELN represents a subpopulation of bipolar cells, and we can see that indeed RELN localizes to a subcluster within our newly reclustered object. This basic reclustering works for most users, but some users may prefer more advanced settings. By checking the Show Advanced Recluster Settings checkbox, we can visualize different dimensionality reduction techniques, such as t-SNE or PCA. We can also change the granularity of which this reclustered object is divided into. So by selecting the color cells by new cluster, we can crank up the granularity to identify lots of different clusters, or we can decrease the granularity a great bit, and this makes fewer clusters. So scrolling up here. We can also change the number of variable features and the selection method of these variable features and renormalization. However, if we adjust any of these parameters, the changes do not take effect until the user selects reclustering, which requires the slow process to restart. Most interestingly, Spectacle facilitates very flexible differential expression analysis. In order to access the differential expression features, navigate to both differential expression tabs. If only one tab is selected, a text box warning will prompt the user to select the other tab. At first, we will compare expression between predefined clusters. They're indicated by this predefined clusters radio button. We previously showed that cluster 5 consists of Mueller cells almost exclusively from the autoimmune retinopathy donor, whereas clusters 7, 8, 9, and 10 consist of healthy Mueller cells. To compare expression between these groups, we use the checkbox inputs to compare expression between groups 5 and 7, 8, 9, and 10. Once these selections have been made, we hit the Start Differential Expression Analysis button. And again, we are prompted with a progress bar that communicates the proportion of genes for which differential expression analysis is completed. While this analysis finishes running, I'm going to take this opportunity to explain a couple of different parameters that control our differential expression. The first is this log fold change threshold slider bar. This log fold change slider bar will only perform differential expression on genes with at least a log fold change difference of the specified value, in this case 0.5. Likewise, the minimum percent threshold slider bar below will only analyze genes that are expressed in at least this proportion of selected cells in one of our two groups. So for example, in this analysis that we ran, the genes have to be expressed in at least 50% of our Mueller cells in 5, or at least 50% of all cells in this selection right here. Setting these thresholds to higher limits results in much faster computation, but less sensitivity. On the converse, setting low limits to these thresholds identifies more genes, but is slower. Here, I left the thresholds as is for Spectacle for a preliminary analysis. After completion, we can scroll down and view the differential expression results graphically and in tabular format. I find this first plot on the left to be very useful, but it takes a little bit of getting used to. Here, each dot represents the differential expression results of a single gene, which can be hovered over. The y-axis depicts log fold change, with positive values representing genes that are enriched in group 1, while negative values representing genes enriched in our group 2 selection. The x-axis represents a variable called delta percent. The delta percent is the proportion of cells in group 1 that express a gene minus the proportion of cells in group 2 that express that gene. So to illustrate this a little more clearly, we're going to look at GFAP, which I can type in the table here and hit enter. We see that GFAP is expressed by 98.5% of cells in our group 1 selection, but only 39.5% of cells in our group 2 selection. The delta percent is 98.5 minus 39.5, which is equal to 59%, which is, as you can see, the value indicated over this hover. What I really like about this plot is that it lets you view the absolute expression level change on the y-axis, while also examining the proportion of cells that express each gene on the x-axis. Differential expression analysis can be made to be even more flexible. If we scroll up, we can define more narrow cluster selections with our lasso tool. The lasso tool results in two plots being populated, and we can identify what makes certain subpopulations unique in their expression profile. So for example, here, these cells in the bottom of this cluster versus the top cluster. Again, we simply re-hit the Start Differential Expression Analysis tab. The differential expression analysis completes, and we can view those results down below. Differential expression can also be performed between cells from different biological conditions. So in this example, I want to identify what's different in cone photoreceptors between the fovea and the periphery. So if I select this Perform Differential Expression Between region, I can draw a lasso tool around our cone photoreceptors, hit Start Differential Expression, and the two identities used for this differential expression are fovea versus peripheral libraries. When I scroll down here, we can see cells colored in red are from the fovea, while cells colored in blue are from the periphery. And we can identify several different transcriptional changes between foveal and peripheral cones. Lastly, differential expression can also be performed on our reclustered object. So if we want to look at other features that make the bipolar cells that express RELN different from the rest of the bipolar cells, we can do so with something like this. Each of these functionalities is supported between the lasso tool and selecting predefined clusters, and they should work seamlessly between the two. So that covers the salient features of Spectacle. I really hope you enjoy using this platform and it propels your studies forward. Thank you for your attention.